Thank you, Beatrice. Let me start with my appreciation for the International Society of Shellfish Immunology for organizing such a wonderful conference, and my appreciation for the organizing committee to invite me to share our research with this group. I have to actually make a declaration that I am no immunologist, so if a lot of immunology questions comes up, I may have to kick it back to you. A year and a half ago, when Beatrice started to communicate, and she asked me to come to this meeting, give a talk, and I was thinking, I'm not an immunologist, but she said, well, we wanted to talk about genomic stuff, so that's probably okay. So with that declaration, I'll share first part of genomics, our genomics research, give you some background, what we have, what sort of platform we work with. Then maybe spend next 25 minutes or so focused on immunological or disease systems that we work with. I have to apologize for two things. One, for those who work with genomics, that you probably hear me talking about genomic stuff before. And also apologize for immunologists that a lot of genomics stuff seems to be very simple, maybe in our views, but seems to be go through very quickly. But I'm sure you are not interested in the specific numbers or in Catfish, but I'll just demonstrate through general thoughts in a way forward thinking to see how genomics can be applied for shellfish immunology situations rather than focus on specific results of our own. Years ago when we started with genomics research, we were facing basically, we were working with catfish. So we specifically channel catfish and blue catfish. Each, we only know that they have 58 chromosomes and roughly about 1 billion base pair DNA. That's what only thing we know. So start from there. A lot of species today, you probably have very minimal or some genomic information, but it's not all that difficult in today's technology to get to where we are today with catfish. The first line of research, of course, we did was to establish genetic linkage maps, which require a lot of DNA markers, but very specifically, we were working with a hybrid system, so channel catfish and blue catfish. So as you can see, channel catfish is pretty good with a number of traits: growth rate, feed conversion, diseases to resistance to columnaris, tolerance to low oxygen. But blue catfish is pretty good with processing yield and resistance to ESC diseases. This ESC diseases system is the one that I'm going to talk about most today. And our goal, long-term goal, is to come up with a synthetic fish that has pretty good traits along all these areas. And to date, we have linkage maps having over 2,500 markers. Still not the highest quality as good as we would wish, but it's pretty good coverage genetic map. The next thing that we have been working on is the physical map of the catfish genome, which is really simple. If you are working with DNA ropes, the idea is that when the ropes are overlapping, you would see the fingerprints of DNA restriction pattern also overlapping. So by using the fingerprints, overlapping patterns of the fingerprints, you can really line these DNA ropes in a linear fashion. Ideally, however many chromosomes you have, you would have that many ropes. That's the best situation. But of course, practically, that's never been the situation because you have gaps in the libraries that break these ropes into pieces. For catfish, right now, we have two physical maps. One that with regular male with about 3,300 contigs. The other with a ganogen fish has about 1,782 contigs. When we combine these two maps together, we have about 2,500 contigs. So that covers the whole physical genome of catfish. The work has been published, so I'll not get into the details. The next thing, when we have the linkage map and the physical map, Clearly, the next task is to get the two maps together. It's quite simple. So we actually just sequence the back ends, then get these microsatellites, then genetically map them to the linkage map, whereby you can really relate the two maps in terms of their interrelationships. To date, we have not been able to integrate the entire linkage and physical maps. We integrate about 52% of the genome. That's because we do not have microsatellites along every context of physical maps, but we are now working with SNPs. Very soon we should be able to completely integrate the two maps. We generated a large number of back-end sequences, a total about 100-some thousand back-end sequences, which I'll talk about in just a moment. That means we have about one sequence one sequence tag per 10 kb in the genome. So if you are working on the entire open ocean, so you would have one sequence tag per 10 kb in the small islands in the ocean, so to speak. Put those islands in the ocean will guide you through the ocean in just a moment as I talk along with the whole genome sequencing. The next area of our major efforts of research was try to identify gene transcripts or gene discoveries. We started with EST analysis for many years. We generated a large number of ESTs. These were standard sequencing, so today it's no longer a big deal. But back then, a few years ago, we were able to generate about 350,000 ESTs from channel catfish, and 150,000 some down here with blue catfish. So overall, we have about 500,000 EST resources from both of the channel and blue catfish species. Very recently, using Genogen, which, as you know, is a doubled haploid, we were able to actually to make full-length cDNAs. We had developed over 25,000 unique gene sequences, identified over 14,000 full-length cDNAs. So this graph shows if, this is channel catfish protein length. That's a reference genome sequences. If the proteins are equal length, you would expect that 45 degrees on that diagonal. That's the vast majority. And in some cases, we have catfish transcript longer, and in some other cases, catfish transcript shorter than the reference genome, as expected. Another major research area is comparative genomics, where we try to have a better understanding of evolutionary events, how evolution, during the evolution, what happened among different species. In particular, we have been working with catfish, zebrafish comparisons, but we also made compare the genomes with catfish, with zebrafish, Medaka, stickleback, and tetradon. These are fully sequenced genomes. As you can see, roughly, very roughly, you have a one-on-one relationship, chromosome relationships. But you also see some other cases where it's not one-on-one relationships. And give you a zoom-in look of the zebrafish and catfish comparison, for instance, you can see zebrafish chromosome number 1, 85% of that genes on that chromosome is located in channel catfish chromosome number 10, and so on. In some other cases, like here, 39% is on here, but there's 18% and 18% on different chromosomes. So give you a feeling, raw feeling, where the chromosome breaks and how they are rearranged together during evolution. This whole genome sequencing was a project that my lab has started about 3.5 years ago. We're almost there, but the thing has not been we have not been able to publish this until probably next few more months. We started with a system that doubled haploid because, as we know, teleost fish has gone through another round of whole genome duplication. So in order to reduce the complexities in the genome assembly, so we made a doubled haploid. So you start with a regular diploid organism. Through this Genogen process, we generated metagenic Genogen that has identical two copies of the genome that would have no SNPs in theory, no SNPs, so you can assemble 100% of the things without any things. And we demonstrated through microarrays that it's identical. So in each case, you see the cell and the dam, but in the progeny, it's all it's homozygous. So the doubled haploid has only one microsatellite allele. So in other words, both copies of chromosomes identical. This project was started in a window of time with the red box. That's roughly in 2009, start there. And it's in a period where technology has really advanced very quickly, and the sequencing cost has coming down dramatically. You can see that's only $10,000 US, 101 million, and start with $10 million and more dollars at that time. So in this window, situation has been very complex. It's a great challenge even for a genomics lab. So we actually started with Sanger, did a lot of Sanger sequencing, 454, Illuminas, and then PacBio. And now we're working with molecular sequencing technologies, which I'll brief introduce in the next few slides. With the channel catfish genome, sequencing has been done earlier, much earlier. So we struggled along with those technologies. Shown in these slides are the summary. So basically, we have generated about 60x with Illumina sequencing, which I will talk about above the red line. We still have PacBio sequences, which covers about almost 10x genome coverage. And we certainly have a lot of backend sequences and so on, which we have not used yet. So far, the assembly pretty good. With Illumina sequences only, we were able to generate about 49,000 contigs, total number of contigs. And in terms of scaffolds, about almost 4,000. But the largest 58 scaffolds account for 50% of genome. 394 largest scaffolds account for 75% of the genome. So pretty good assembly. And we know that about 80, a little bit over 80% of genome are quite unique. And there's 14% of so are highly repetitive, which is very difficult to put together, as I will talk more a little in just a moment. The overall assembly strategy so far, we only use Illumina sequences, which really is down here. And then you put into very short contigs. But very soon, we're correcting the mistakes with PacBio right now. So we will be able to put this PacBio 454 at long reads, then back-end sequences, eventually through the physical map and linkage map, put into the whole genome fashion. So that's not too much different from any other species. However, we did learn a lot of lessons in such an experience. When we started with blue catfish sequencing, we were much better prepared in the last year, saved a lot of money. Partly through the channel catfish sequencing, we learned that the reason that we cannot put together is mostly because repetitive elements. So for instance, here, it's a contiguous sequence DNA from very continuous, but you have two stretches of repetitive elements in this example. Because it's repeat, so computer software has no way of knowing it's coming from here or it's coming from here. So as a result, the assembly would generate 3 contigs there. That really leads to 2 unwanted consequences. One, your contigs would become smaller. And second, these things are falling behind as sort of trash or garbage sequences. They're packed on there that you cannot assemble them together. So we learned a lot of these things, so we tried to use a different technology, that molecular technology, which molecular long reads really still uses Illumina sequencing, but it's clonal sequenced. So when it's clonally sequenced, you can see now, for instance, this piece of DNA, originally the long stretch of DNA has repeats, but now with this clone, you only have 1 repeated area. Another clone has this repeat, so as a result, within this clone, you can assemble them together because all the red now you come there, and within this clone, you know the red comes from here. After you do the clonal assembly, you do the second assembly that will allow you to put all this continuously, put the sequences together, which is really neat in a way. So it's Illumina sequencing, but clonally sequenced. So in this regard, you can see the read length is very good within the molecular sequencing. Here we have about, that's over 3x genome coverage. The average within clone read length is 4.6 kb. So that's for the most part run over the repetitive elements. In Catfish, the vast majority of repeats are TC1 Mariner transposons, the size of which is about 1 kb, 1.5 kb, 1.6 kb is a full length. So give you a sense, for the vast majority, you can go through the repetitive elements and assemble the whole genome together. So here is what I always say. Now we have many fish that genomes are sequenced. Many shellfish genomes are sequenced. But we have to, as genomicists, we have to have a general sense. In a way, genomes were always known because you know genomes in Catfish has 1 billion of ATGCs. The matter that we do not know is how they are arranged together, how they are combined together. After genome sequencing, in a way, you organize these sequences ATGCs into these short tags. But it's still in a pool. You have no idea of knowing what is the interrelationship of such pools. In our case, we still have 49,000 contigs, which is not bad, by the way. Compared to Turkey genome, it has 159,000 and so on using next-gen sequencing. But this is not good enough. So we need to link them together with other resources that talk about like long reads, back-end sequencing, and so on. Of course, everybody, when we get started, we think that this is what we're going to get with the genome assembly, a beautiful castle to start with that dream. And I hope that we try to avoid something that eventually we're going to end up with this, then that's certainly not good enough. So we made some assessment to see how we're doing. I think that we are doing fairly well in terms of these parameters. So 4 parameters are very important for genomic sequencing. Accuracy, in other words, ATGCs, if you make a mistake in the columns. Completeness, what percentage of genomes are sequenced. Continuity, the contig sizes, how big is it? And finally, connectivity, so relationship between those contigs. These are very important. So in blue catfish, we feel good about it in all these 4 parameters. In channel catfish, accuracy pretty good, connectivity very good, but we are working to improve completeness and contiguity right now. This slide is really for, like Antonio, Dr. Figueres yesterday talked about the dark side. This slide is really for the dark side. I myself also associate dean for research, so I sort of understand both arguments on both aisles, both sides of the aisle, that aquaculture obviously is very, very important. It accounts for $160 to $180 billion of the world economy, very, very important. And it's the only agricultural sector that grows in 5 to 7% annually in the last 20 years, the only sector. Yet, diseases challenge is a huge challenge. And I do not know how many administrators are sitting in the room. If you are here from Europe or States or anywhere, China or anywhere, make sure you pay attention to diseases research for aquaculture. Make sure you invest dollars into this area. And when people sit in this room will make a difference. It may not be, individually, one of us will make that a big difference, but collectively, this group will make a difference. So invest into this area is a good thing to do. And I'll now switch turns a little bit, give you some detailed examples how we apply genomic resources, genomic approaches for diseases research and for particularly innate immunity research. So the first area of research is with the genomic resources, we can really systematically analyze immune-related genes. And you get all the pools of gene sequences. You can do the phylogenetics and so on, and study where they are expressed, in what tissue, what stage, that type of thing. Then verify with real-time PCR, mostly in recent years, before and after infection at different times. Get a sense. Generally, that belongs to, I call that a functional correlation. Basically, if a gene is important, it probably will be up or down or have some changes in that process. If it's have absolute no change, you cannot exclude the function of that gene in the diseases. But if they are way upregulated or way downregulated, probably somehow they are related with diseases processes or particularly defense responses. So that's the approach we have been using. Here's an example that, for instance, very recently we identified and characterized 20 TAR genes from catfish. And by compare with other species, you can see that some are only in catfish, like this TAR 2526, found only in catfish. And some are only in fishes, like this 18, 19, 20, found only in fish. But some are not in the fish, like this, this blue block that were not found in the fish at all so far. But in a number of cases, we know it's highly likely they are not in fish because we have the whole genome sequence. We have pretty good confidence in terms of how those things evolve and so on. Now, how they are in fact involved in the disease resistance, and functionally, in terms of correlation, their expression here generally upregulated is whether the TLR genes involve these ones or RLR or NLR receptors, they are generally up in the spleen and liver, but in most cases down-regulated in the head kidney. We do not quite know why, but people in this room probably can easily tell me why. Probably involves some of cell migration from tissue to tissue and the like. So in this fashion, we have worked with various gene families, including antimicrobial peptides, the chemokines, cytokines, TLR, and other receptors, leptins, lysozymes, complement systems, all the factors and inols, all the related proteins such as ferritins and keraticulins, and so on. In this analysis, our hope was trying to find some markers that are related with resistance and susceptibility. You can see the genes shown on the left panel, that such as lysozyme C, inols, MHC1 pathway genes, interleukin-8, TNF-alpha. These genes are related with resistance generally. Higher expression of these genes are correlated with higher resistance. The genes on the right, that ribose-binding lectin, interlectin, mucin, and so on, these genes, high expression of these genes are correlated with high susceptibility in catfish under bacterial situations. But we're trying to find a better approach, but this, as much as we can so far, correlate the resistance and susceptibility. A few years ago, Eric Pittman was my former PhD student. He is now in our faculty at Auburn. That he started this microarray research. Basically, that we are using the hybrid system. Again, channel catfish is susceptible to this bacteria of EST. Blue catfish is highly resistant. So we were interested after challenge to see at different times what happens in terms of gene expression. So of course we found many, many differences, and shown here is one simple example of 5x or more up-regulated genes. Clearly you can see 3 in 3 replicates here without infection. After infection is shipped here, many genes are way up-regulated. On the right is a blue, very similarly 3 replicates highly regulated in here. But you do see differences between China and blue catfish. Particularly MHC1 system is much earlier. It's activated much more early, much earlier in blue catfish than in China catfish, which could be account for the observed resistance, correlated at least. We do not exactly know. But the major problems on microarray studies is the inability of researchers. I always challenge my students. I say it's not the technology, it's our brains. The researchers, how smart we are, how well we interpret the data is a limitation. So I think I call that listomics. You generate a list, but so what? That's the limitation. So very recently, we started with another line of research using RNA-seq. The technology is very, very straightforward. You just basically get the RNA through next-gen sequencing. Then you align your reads, short reads on the reference genome or reference transcriptome in our case. Then you compare before infection, after infection. In this case, for instance, if this gene were sequenced 6 times before infection, 18 times after infection, you know, oh, this gene was upregulated 3 times, so, but calibrated somewhat by RPChem, so reads per kilobase per million. So from X to Y, you can calculate that ratio pretty easily. So we have published a few papers along these lines, and the technology works really well. And in this analysis, we actually had some really surprising findings, particularly like in this case, cytochrome P450 genes that we very minimal reports in the past, but many of them are highly upregulated after infection. For instance, in the slides, the 10 cytochrome P450 genes are upregulated, some extremely highly upregulated, like over 30 times of this one, 20-some times and so on. And we also found a number of cytochrome P450 are downregulated after infection. Many of them are very, very highly downregulated. One of them is downregulated over 50-fold there. So clearly, this technology is very sensitive, allow you to detect any genes up or downregulated, no problem. Another line of research, we have been developed a 250K SNP array, which really allow you to do a lot more than just to see up or down regulation, because now you can look at the genotypes. We have been using the Affymetrix axiom technology, which is pretty good. And here are the 250K array. So we have over 146,000 from channel catfish. These are from genomic regions. Then from genes, we have 32 samples, 31 from both channel and blue. We also have the interspecific SNPs, almost 40,000 there. Total 250,000 SNPs. In other words, we have 1 billion base pair genome. So think about this 250K SNPs. So we would have 1 SNP on average every 4 KB. So the coverage is really pretty good. And of course, the spacing is a major issue. That the perfect scenario would be if you can have a molecular ruler. So you have put 1 SNP every 4 KB. But practically, that's almost impossible because 1, it's limited by the availability of SNPs, and 2, we do not really have a full assembled catfish genome yet when we designed this array. But here is a rough assessment. When we put these SNPs, gene-associated SNPs onto zebrafish genome, you can see it's distributed pretty good along all 25 chromosomes. And along the chromosome arms, in some cases, like chromosome number 4, you have pretty low levels of SNP there. But nonetheless, you have plenty of SNP to select from to build that array. And very recently, we tested this array. The converting rate is over 80%. These are profiles of SNP genotypes. The top 3 are good ones. These are bad ones. So adds up those 3, over 80% conversion rate, so pretty good. In other words, 250,000. Now we can convert over 200,000 as good SNPs. So that's pretty good in a sense. So with this array, we are now able to do some GWAS whole genome-wide association studies. So in our system, we recall that I keep saying that we have channel which is susceptible, blue catfish resistant. We have F1. We have backcross with the susceptible parents, give you this backcross, F2 generation backcross progenies. Then we can ask what genes are associated with the diseases resistant in this case. Here are some initial analysis. This is linkage group 3. You can see there's a long segment from roughly 15 megabase to 36 megabase, highly associated. Those are p-values over 10 of the minus 10. So pretty highly associated with linkage group 3. Here another one with linkage group 5. This is really actually sharply, the LD is sharply decaying. You can see very high p-values right there, I mean very low p-values, over 10 of minus 10, but very quickly decaying here. So some genes right around this region would be important for disease resistance in this case. One of the gene is TLR20, located right there. We're not sure which gene is the gene that is important, but clearly in that region, there's a highly associated disease resistant gene in chromosome number 5. This chromosome number 13, very similarly, you have very high association, SNP association right there, then very quickly decay in here. So these chromosomes apparently are very important. We also, because we still have many contigs that are not associated with the chromosome, so some of them are highly associated with resistance. For instance, in this region, you can see that that's fairly highly associated. We initially thought this would be related with this one, microglobulin, but it's not. The most significant SNP is not in that gene, but that gene happened to be right there. Similarly, with columnaris, we think that the two particular linkage groups, linkage group 7 and 12, are very, very important. So next strategy we have been doing is we call the BSR-seq, so bulk segregate RNA-seq. This is a very neat technique. Many years ago, back in 1991, Richard Mitchell at UC Davis started with this bulk segregate analysis. We combined that with RNA-seq. In other words, you have the fish, then you can actually challenge them, then you have the earliest mortalities, and you have the survivors. Now you can have these two bulks. So the two bulks represent then the susceptible fish and the resistant fish. Those we call bulk 1 and bulk 2 in the next few slides. Clearly, with BSR-seq, you can, like RNA-seq, you can very easily to study genes up and down regulated. In this case, within susceptible fish, you have over 771 genes up, 400 some genes down. And very similarly, in resistant fish, you have 130 genes up, 94 genes down, and so on, which is a very traditional RNA-seq that can do. What's important is that 57 genes are uniquely upregulated in resistant fish. So when you compare resistant and susceptible fish, clearly you can see the differences. So these genes on this side, each dot represents one gene that upregulated in resistant fish. On this side, upregulated in susceptible fish. So we were also trying to see what really gene pathways are involved in here. When you look at, actually recently, in collaboration with Dr. Enchley, working with shrimps, we found the proper activating system is really working a lot in the shrimp system. But when we locate these genes in the catfish situation after bacterial infection, you can see those genes involved in proper system is also activated. And I showed here the 4x, which is a BFR, so bulk frequency ratio, which I'll explain in just a moment. But very similarly, also many genes involved in endocytosis clearly are differentially regulated in resistant and susceptible fish. I will not go through the details because this is a lot of details, but just tell you that there are different segregation, there are different activation, and there are different allele usage among the different groups of fish. Very similarly, in the complement pathway, we also have gene segregation differences, regulation differences, and allele usage differences. So here, I'll explain a little bit of bulk segregated RNA-seq in a way. So we use this BFR quite a bit. So suppose genes are at work. Then here, resistant, you get most A's. Hopefully, in the susceptible, a different group of fish, you see mostly G's. So in other words, the choice of alleles is very different. So caused by genetics, which is what we're most interested in. So in this case, we are interested in this BFR, so bulk frequency ratio. So here, for instance, you have 11 to 1, white to yellow, and here, 1 to 11 in terms of this white, so the BFR would equal to 11. So when you have BFR large, like equals to over 10, that's highly differentially allele usage between the two groups. And in this regard, we identified over a million SNPs, and some of them are significant. So like 56,000 SNPs are significant. That involve genes, that 4,000 genes. And when we look at large BFR, greater than 4, 359 genes are larger than 4x, which is that red one. Some are extremely large, over 16x. In the example shown earlier, that's 11x. So give you a sense how serious. When you look at this BFR only at the gene expression level, you see different genes. Now you see this microglobulin show up again, complement, and apolipoprotein B shows up being very, very significant at these peaks. But most of these situations are caused by gene activation, not by genetics, actually. So when we look at genetics, we ought to look at situations look like this. So in the resistance, now you look at the DNA level and RNA level. So you will see at DNA level and RNA level, ratios are very, very similarly. So that means pretty good. So that means it's caused by genetics, so in this case. And, but when you combine these things, so in here, for instance, this first bulk, now mostly A, but you have one yellow, and here mostly G and one A. But when you combine these two pools, now you get 1-to-1 ratio. This means that these are caused by differential allele usage. In other words, caused by genetics that our lab is mostly interested in. When we look at those segregation patterns, now you can see the red dots here is low allele ratio when you put them together. So resistant and susceptible put together, you have very, very low allele ratios. Those are the ones that we're most interested in. But you also have some other that allele ratio are very large, and yet BFR is very small, which is caused by gene segregation. When we look at genetics, we have many significant SNPs. We ask where are these SNPs located, or where are these genes located? In this case, in linkage group 3, recall that has resistant genes, and that in this region, very significant BFRs, so over 30, 40 BFR. So one of the genes is tumor suppressor candidate 5 homolog. We do not know that's the gene or not at this point, but clearly it's related with resistance. There's another linkage group 6 here, this protein G7C-like gene, very, very highly significant, over almost 20 BFRs. Linkage group 13, very similarly, in this region, involve interferon-induced very large CTPS1-like gene that have over 20 BFR, so very, very significant. So another level beyond genetics is at the gene expression level. In other words, you see DNA at the DNA level, the allele ratios are very similar, but you see very different RNA allele messages, which means one allele get activated or get induced, expressed only. In this case, for instance, you see these are very similar, white and yellow, but here only white allele get induced a lot, which is because of gene expression. So again, when we try to ask what are these genes, some of them, this red circle, these a few genes are caused by both. In other words, they are segregated differently at genetics level, but the green spots up here above this 14, above this line, is mostly induced by gene activation or differential allele specific expression rather than allele segregation at the genetics level. So when we look at these things, we found different things. We still do not know exactly which of the origin this comes from, but in some cases we do know. So these red triangles represent those alleles coming from channel catfish, and the blue triangles represent those coming from the blue catfish. Many we are working on, we do not know yet. But clearly, this leads to us to think about one thing. It's not only genetics is important, but also gene activation is important. The third thing, which we have never thought about, maybe the allele combination. You have different alleles for under resistant situations. You have from different genes, different alleles give you the best combination for the best resistance. So I think that's a new area that we should really invest a lot of energy to study. So in summary, this BSR-seq allow you to do really three things. The first one, of course, like RNA-seq, that allow you to identify differentially expressed genes that leads to expression candidates. Second one, that allow you to identify SNPs and significant SNPs eventually give you positional candidates. And the third one, particularly, I think we should pay a lot of attention. It will allow us to identify specific allele expression. So which ones and which combinations give you the best resistance. So this slide is the last slide, but I use a lot in my seminars recently, and I hope we really bring attention for the aquaculture geneticists and immunologists and the like. These are the chickens in the 1957 and 2001. You can see 43, day 57, 91, 71, 85 days after birth. You can see the difference. So 50 years ago, 50 years later. So if poultry geneticists can make such a difference, and I try to inspire every aquaculturist that if we work together, we can make similar impact to the world. So with that, I'll stop to take any questions.